So I think we're ready to start session 6, and the chair for this session is Lou Cantley from Weill Cornell Medical College.
Hey, welcome everyone. It's a pleasure to get a chance to discuss in this session nutrition. We have 3 speakers, Joanne Kelleher, Bruce Spiegelman, and Jin Gu. And Joanne, I should say we're going to have some transition from the previous session in that Bruce Spiegelman will talk quite a bit about exercise as well. But let's start then with Joanne, who has spent much of her career working on isotopes labeling in nutrition, and is really a pioneer in this area. And she will tell us what she's learned.
Hi. So I'm here to start this nutrition session, and I thought I would distinguish between 2 themes that one may have in nutrition in this presentations that we've had at this wonderful meeting. And one might be a cellular physiology theme, where we look at genes, transcripts going to fluxes, and as Christine DeRosier, my colleague of many years, pointed out how important they are, and then their cellular mechanisms. And this is very important, and we've learned a lot of neat things about this during this meeting. But I'd like to try talking about another theme, going from food to digestion to transport to metabolism, and then to cellular mechanisms. In other words, we're coming from the other way around. And we'll see how that might add something to the plans for improving human health. And to do this, when I first started this, I asked myself, why does it? I don't see that. Yes. So what's the big problem that we really need to discuss? And for me, a big problem is chronic diseases of Western man. This is the enormous out of control problem. And how do we think about this? Having gone to many meetings as a cell biologist and metabolism person, seeing all sorts of fancy displays and the newest and latest drugs, and each one seemed to fail a few years later, and we just seemed to get worse and worse. And then I came to read a lot more about this as an older person, and decided the solution to this is simply to change the diet. And that's easy to say because it's hard to do, just like the exercise we discussed earlier. But let's just talk about this importance of the diet on this whole issue of diseases of Western man. We all know about this, but I think we don't always apply it as much as we could. And I've been inspired in my reading on this by T. Colin Campbell at Cornell in his wonderful book, Whole, which is goes into much more detail on these sorts of issues. But the whole food plant-based diet is clearly a way to improve our health. And what is the whole food plant-based diet? It's no meat. We try to minimize meat, dairy, eggs, fish, and processed food. It's not exactly the same as a vegan diet, but it's a diet that can be shown to lead to very healthy outcomes. And more about this is available on the website cited here. And if you look at studies in nutrition of diet and health, one of the best ones I think is the Adventist Health Study, because these are people living in the US with the same lifestyles most of us have. And when you, in this population, of course, you have vegans to non-vegetarian people who eat pretty much the standard American diet. And when you look at their BMI and their risk for any chronic disease, here's type 2 diabetes. You see it follows, of course, that pattern. And that's very interesting. It tells you 2 things. 1, this is the best diet. And 2, that making small changes can be very helpful. Moving in this direction towards the healthier diet can be very helpful. So with that in mind, of course, you'd ask yourself, well, is it just having a lower BMI that causes the good health issues? And for many other studies, and here's the Women's Health Study here up on the upper left that I would cite, they've shown that clearly even when you adjust for BMI, There's a positive association between intake of red meat and processed meat and the risk of type 2 diabetes. So the risk is not just, that low BMI leads to less diabetes, but also lower consumption of red and processed meat. And this is borne out in many other studies. So, and you all have seen these, I'm sure, many times. You can look at other diseases such as colon cancer. Here's the dramatic changes in incidences of colon cancer in rural Africans compared to African Americans. And of course, non-African Americans are up here too, much higher than rural Africans. When you look into the difference in the diet, what is it that makes these rural Africans so much less inclined to get colon cancer? The biggest change is they have a lot more carbohydrate in their diet. And it's very important to emphasize that a whole food plant-based diet is a high carbohydrate diet. It's a low protein, low fat, high carbohydrate. But the carbohydrates are complex. They come from grains and beans, not from Wonder Bread, et cetera, that we tend to have in our Western diet. So the carbohydrates are an important change in the diet, and also, of course, the enormous amounts of fiber that come in these diets that are in the whole food plant-based diets have huge amounts of fiber compared to the standard American diet. So these are things that show us over and over again the importance of diet. And although I agree with everyone that it's very hard for people to change their behavior, having teaching a nutrition course for undergraduates at MIT, I've learned that undergraduates are very, very interested in this. And we need to make sure that our brightest undergraduates have access to nutrition courses that teach them about these issues. They're obviously very concerned about climate, sustainability, population growth, and health concerns. And so all of these things will help push this tipping point to away from our standard diet, standard American diet, which we call the SAD diet, towards this whole food plant-based diet. This can happen, and the young people will be doing this. So the next question is, why are whole food plant-based diets best for humans? Well, there might be three reasons. One is processed food is clearly toxic. Two, there's this evolutionary history we have for many, many eons, well, not eons, but for a very long time, our ancestors ate a largely plant-based diet. These gorillas eat largely elephant grass. They can eat 45 pounds a day. They get all their proteins and nutrients from this. It's a perfectly healthy diet, all plants. The third reason is that plants are evolved to deal with oxidative stress, which is a major factor in diseases. And they do that by producing lots of interesting molecules, carotenoids, polyphenols, et cetera, that fight oxidative stress. And obviously they have to balance the needs, the amount of oxidative and antioxidants that they have, but they are very acute at doing this, and they provide these antioxidants to us. And since oxidative stress is a major factor in our diseases, no wonder plants are good for us. But now we ask, how do we get from knowing that plants are good for us to mechanisms? Here we have sort of a state of knowledge, and we know a lot about cellular physiology and almost nothing about, and a lot about holistic human nutrition, but it's very hard to link between these two things. It's very hard to figure out just exactly what mechanism is involved when we see these clinical studies. So we need something new. We need new studies that they must be holistic. They're going to be big, messy data sets, and we're going to have to figure out how to deal with that. Many approaches are needed. My approach is via stable isotopes and mass spectrometry. So I want to make heavy food using deuterated water. From deuterated water, you can make heavy tofu, food, eggs, dairy, etc. We want to make this heavy food with deuterated water and then back calculate from a subject where his leucine, for example, here came from. Did it come from his own body that he already had before we started the experiment? Did it come from the tofu that he ate? And did the tofu's leucine come from the deuterated water or other sources? And we have some expertise in doing this. We also have expertise in non-targeted mass spectrometry to look at sources of labeled compounds in using mass spectrometry. And combining those two things, we're now interested in these newer studies where we would look at plasma samples, feed people deuterated food, look at plasma samples. Again, try to use our own skills plus some AI assistance to figure out what molecules, what peaks come from food, what come from endogenous sources, and what are something new, what molecules were made maybe by gut bacteria from food that we ate yesterday, et cetera, and this kind of thing. So once we could do this with deuterated things, perhaps we can get AI to help us take the deuterium out, and can we do it with food without the label in? And these metabolites that we're interested in, of course, are all of these huge number of polyphenols and carotenoids, et cetera, that are in our diet that are helping us fight antioxidant stress and other trials in modern life. We'd also get 4D information here. As you see, when you put the label in, you'll get some information about kinetics. So at the end of doing this study, of my heavy food study, I think we'd be ready to present to the 4D cellular physiologist concentrations of metabolites right outside your cell right now, and you can tell us what happens when they go inside the cell and how they work to change the metabolism and promote health. And that's it. Okay.
We have time for a few questions. There's one from the audience. From Philwa Lee. Wonder how GMO foods are digested by the microbiome.
GMO foods, I don't think I know of any way to tell. I don't think GMO foods, I don't have any comment really. I don't think it's going to be any different really.
Okay. So, one question I have is about the difference between fruits and vegetables, and particularly the increased fructose in fruit, and whether you're trying to look at particular types of ingredients in the food. You could clearly do that with isotope labeling, at least by pulsing it in, as to whether different carbohydrates are processed differently.
Yes, of course. And you could imagine adding this and then dissecting this out. The first level of this is a non-targeted approach. But clearly, once you have a plasma sample and you've seen something in a non-targeted approach, you can go in with high-resolution mass spectrometry, perhaps with other tracers in combination, and do more elegant studies. I think the thing about fruits is, traditionally, fruit is very good for us, and the sugar in fruit doesn't seem to be terribly bad for us. But now we've made these fruits, like we make grapes sweeter and sweeter and sweeter, and we've engineered our we call whole foods to be something else. So it can be an issue, and certainly it could be explored. Yep. And, you could make Wonder Bread, heavy Wonder Bread, and look at it too if you like them.
Yes, good. Okay, I think we're near the end of time for this talk, so let's move on to the next, Bruce Spiegelman. Bruce?
Yeah, so I guess I need to share my screen.
Yeah, I think very good.
Okay. All right, how's that?
We see it.
Okay, good. So thank you for organizing this nice meeting. I found it very interesting and useful, and I'm interested in both sort of molecular nutrition and exercise and adipose tissues and muscle. So I thought I'd give a talk about something that actually Deb Moyo related to, which is how should we approach exercise. And so I thought I would discuss how to capture some of the healthful bets of exercise in molecular form, and the methodologies that we might use to do that. So I'm going to talk a little bit about that and then just give one example. So very important, and this is something Julene referred to, we're meant to move. And I will also point out that evolutionary biologists have pointed out that the increase in size of the human brain correlated very tightly with our being a more migratory species. And presumably, when you migrate more and you investigate more environments, that involves both increased movement and being better at moving, and also being better at thinking about your new environments, cognition. Physical work is part of the whole body energy balance systems. Obviously, this is on the energy expenditure side, so it has consequences in the whole energy balance problem, physical movement. So this is, I think, a critical thing. I think most of us already know this. What are the health benefits of physical activity? There's probably, most of you are familiar with the effects on cardiovascular and type 2. You may not be aware that this is the strongest correlation with defense against dementia is physical movement. The odds ratio in the chances of getting Alzheimer's are greatly altered by whether you were sedentary or physically active. In a walking program in adult middle-aged humans, physical activity will increase the volume of your hippocampus as opposed to the normal age-related decrease in volume of the hippocampus. The last thing I want to point out, again, because this isn't as well known by the general population, physical activity reduces the risk of colon cancer, breast cancer, several other cancers. And it isn't just by reducing obesity, although that is a component. That exercise itself has an effect on cancer incidence that is separable from the effects on obesity and energy balance. So this is a big array of interesting results obtained over the last 30 years. Now the question is why? What are the molecules? That's what interests us. So we are particularly interested in, have been particularly interested in the idea that muscle might secrete stuff that affects all these different tissues. And how do we think about isolating and studying the stuff that might get released by muscle in the context of exercise that might impact these other tissues? There are models of exercise both in rodents and in human beings. Mice are very easy to get to run and swim, which we would characterize as endurance exercise. There are modalities being developed now for resistance training in mice, although that's still a bit tricky. It's actually, exercise is one of these unusual things that are actually easier to study in a human than it is in a mouse, because resistance training, endurance exercise, all these different modalities of exercise have been very well worked out in human beings, a little bit less well worked out with mice. So here is a sort of broad overview of how can you study molecules that are produced by muscle with exercise. Well, sort of the new old-fashioned way, it's not so old-fashioned, but it's pretty old-fashioned now, transcriptomic plus predictions of secretion. This is low-hanging fruit. Anybody can do RNA-seq on muscles under different protocols, in mouse from human, apply some prediction tools and figure out who is likely to be secreted, although those are imperfect tools. Another approach, which I have to take exception to something that was said very early in the meeting, that we want to get away from cultured cell systems. I would argue that cultured cell systems are still very much appropriate for certain things, including this, proteomics of secretions from cultured muscle cells under certain stimuli and genetic manipulations is still highly relevant. Something that we are more interested in and excited about in 2021 is proteomics of interstitial fluid. So we've developed some methods along with my colleague Ed Chowchani to specifically isolate interstitial fluid from muscle and fat, sort of get the stuff between the cells, and it can be obtained in pretty good form. And so then this allows then proteomics, a high resolution mass spec of both small molecules and large molecules to interrogate these fluids. Now, holy grail, which we are not there yet, is proteomics of plasma. Right now, it's a disaster. I mean, it's because of the necessary requirements, 7 orders of magnitude, and the abundance of different polypeptides in the blood. You can't really realistically do mass spec of plasma today, but that doesn't mean that we won't be able to 5 or 10 or 20 years from now. Metabolites in the plasma studied by mass spec. This is something we just heard about. Yes, this is something that can be done with plasma with quite good resolution. So let me give you my example. So in 2002, we put this molecule called PGC1 alpha, this transcriptional coactivator that we discovered a few years before. We put it in muscle. We thought we were going to study mitochondrial biogenesis. And lo and behold, we got an exercise, endurance exercise-like phenotype, quite a severe one. It involved angiogenesis, anti-atrophy, anti-dystrophy, fiber type switching, beta oxidation in the muscles. Very robust and broad exercise-like program. In fact, even a little extreme. It does these things more severely than would ordinarily be seen with exercise. But we thought it was interesting that maybe this would be a model system that could be used to capture some of the basis molecular benefits of exercise, in the sense that only one tissue is really changing in our model, the muscle. Trouble is, when you run mice, many tissues are doing many things. Here we could isolate one tissue, cell and culture also. So, we and other people over years showed that, in fact, PGC1 alpha in muscle recapitulates many of the effects of exercise way beyond just beta oxidation, mitochondrial biogenesis, and anti-dystrophy, anti-atrophy, angiogenesis, fiber type switching, neuromuscular junctions. So a severe model system, but a model system nevertheless. And so we use this in subsequent years to study what muscle is making under this exercise-like perturbation. And we discovered a molecule that we named irisin, after the Greek messenger goddess that goes between Olympus and humans. We didn't want to name it for any particular function, so we named it basically for its messenger-like function. We studied the browning of fat initially, but we've studied a lot more things since then. So this molecule irisin is a membrane protein, highly enriched in muscle, skeletal muscle and brain. It gets cleaved, proteolyzed, released into the bloodstream, and then presumably goes to many other tissues. We showed, people thought this was weird and it was probably an artifact, and we showed eventually using high resolution tandem mass spectrometry that irisin does in fact increase. It does circulate in humans, it increases with exercise, and it circulates at typical hormone-like levels, 3 to 5 nanograms per mil. And I'll give credit to Lee et al., who before our work had already shown that submaximal exercise in humans, these are patients before and after exercise, causes an increase in irisin levels in their bloodstream. We published the receptor a few years ago, alpha V beta 5. An integrin is a major receptor, or we don't know that it's the only receptor, but alpha V beta 5 is a major receptor done by Chad Kim in my lab using osteocytes as a target cell type. And here's what I want to leave you with. It's pretty exciting. So a number of papers have come out over the years that exercise helps our brain. We all understand that. Irisin may play a role in this. This was actually a New York Times story about a different paper, not our paper, but by a Brazilian group who works on this now. But the one thing I will say about that is, until recently, we had played with FNDC5 genetically. The Brazilian group had played with FNDC5, the parent protein from which irisin is cleaved. And just now, we have a paper coming out in 3 days where we show that actually irisin is the active moiety. And particularly, we tie it into cognition such that a mouse that lacks FNDC5 irisin will not get cognitive improvement with exercise. And furthermore, in 4 different animal models, including wild-type mice, if you elevate blood irisin, you get improved cognition. In some models, markedly improved cognition. And I'll just show you one example. So the methodologically, this is important. We introduce it into the blood. The way we do that is use an AAV8 that's taken up by the liver. It expresses cleaved irisin. It doesn't get taken up by the muscle. It doesn't get taken up by the brain. And so as far as we know, all we have done is elevated irisin in the bloodstream. The Morris water maze test, some of you may know, you train a mouse how to get out of the water because it would rather get out of the water than swim. Then you put like a milky color in the water so they can't use external cues very easily within their little tub of water. And they learn, this is a memory test, they learn where that platform should be in quotes, to get them out of the water. That's the Morris water maze test. Yeah. And lastly, that when the mice get irisin, this is a model of Alzheimer's, and when the mice get irisin in their blood and it's elevated, they get much smarter at remember where their escape platform is supposed to be. And so, as I said, this is coming out in 3 days. And this allows us to say that this irisin molecule made by muscle with exercise is at least a good chunk of, if not the connection between cognitive ability in mice and their exercise-like behavior. And I think there are many more things to discover. As a biochemist, I find this very exciting. I think there's a lot to be discovered, not just about the molecular basis of exercise, but what muscle is putting out into the body to talk to these other tissues. With that, I'll wrap up.
Okay, thank you, Bruce. We still have a few minutes for questions and discussion. I don't see any in the Q&A yet, but I'll start this off by asking, could you give a little more detail on the molecular mechanism by which this gets cleaved and released from the muscle, irisin?
Good question. Not too much is known right now. It is made as a membrane protein. The membrane protein is exercise dependent. And then it has a pack of basic residues in the juxtamembrane domain. So it's a type 1 membrane protein with the irisin part sticking into the extracellular space. And then a molecular invitation to be cleaved, where I think you have 5 out of 7 of the residues that are juxtamembrane are basic residues. So it's going to be a trypsin-like cleavage, but specifically the protease has not been discovered yet. And whether that proteolysis event itself is regulated hasn't really been published or studied yet. We've been just busy with these other aspects that I've been mentioning. But it's a good question, and it would be super interesting to know whether the cleavage and release is itself regulated. You don't need to do anything special. If you express FNDC5 in almost any cell in culture, some of it is going to be released. So, but it could well be regulated, but I just can't tell you what that regulation is.
Okay, let's see. I think we do have some questions here. From Maria Fernanda Forney. We know that some aspects of endurance training can be mimicked by calorie restriction. There are myokines that are produced in response to both exercise and calorie restriction that are produced in global shared response to energy deficit.
That's a good question. I have no idea. It's probably worth studying. I believe it's so. Irisin, FNDC5, is downstream from PGC1 alpha. If I'm not mistaken, it's been published that caloric restriction actually induces PGC1 alpha in muscle. So I could guess that the answer might be yes, but we certainly haven't studied it, and I'm not aware of anybody else who has.
Okay, another question. Is it possible that this is from Kartik Rajagopalan, I think I got it right. Is it possible that irisin might also improve cognition in non-Alzheimer's models like wild-type mice?
It's in our paper. So yes, the answer is yes. It improves cognition in wild-type mice. We show that in the paper that's about to come out. And the reason we did this in several different models, for those of you who are not aware, as I was not aware, the Alzheimer's field, whatever model you use, they're gonna dump on it. They're gonna say you used the wrong model. There's no consensus at what the right model is. So our way of dealing with this in the current papers, we used 4 different models, 2 AD models. The FNDC5 knockout itself has cognitive deficit, and then wild type mice. So yes, I have to think there's no reason to think this is just AD. I think, in fact, I don't think this evolved related to AD. I think, as I said, evolutionary biologists seem very firm on the concept that the ability to move was linked to the size of the brain and to cognition. And I think this is, the data suggests that this is one of those links.
Okay, I'll take one more question. There are a couple more that you can go answer yourself after this, Bruce. Padma Maruvna, is there a dose response time dependent response to irisin on cognitive improvement?
There's no reason to think there wouldn't be, but these experiments were so long and expensive that we picked a dose that in preliminary experiments looked like we were getting positive results, and we just went with it. We just don't have the bandwidth and the expense to do this as a proper dose response. I assume if the work keeps going in our lab and others, this will eventually be done and dose responsibly. But this was 2 years and a lot of money to get these studies done. And so, yeah, dose response is important. We haven't done it yet.
Okay.
I will say, it's not that we didn't do any doses. We did little experiments to find out what was working, in quotes. And then that's the dose that we applied in bigger studies across 4 different animal models.
Good. So you can look at the Q&As to these other questions, but we can bring these up again also in the general discussion at the end. So I want to move now to our next speaker, which is Zhen Gu from David Sabatini's laboratory, who will be talking about the role of TOR in response to nutrients.
Hello, can you hear me as well?
Yeah, we can hear you and we see your slide. Needs to go into presentation mode. Good.
Yeah.
Thanks.
So, hi everyone. My name is Zhen Gu, a graduate student from the David Sabatini lab at MIT. I'm very excited to have this opportunity to share a very interesting story from my point of view with all of you. It's very related to the regulation of the food choice by the mTORC1 pathway. And it's a collaboration with Patrick Joanne, who's a postdoc from Nobel Paramount Lab at Harvard Medical School. And my lab has been focusing on trying to reveal the upstream regulation of the mTORC1, which is a very essential protein kinase complex that responds to various environmental cues to regulate anabolism and catabolism, and eventually impact growth. And from an organismal point of view, we think mTORC1 actually serves as an antenna to process the information provided by the nutritional value in the food to regulate growth. And amino acids are actually key components of nutrients. And among all the 20 protogenic amino acids, leucine is a very potent activator of mTORC1. And that's one of the main reasons that the bodybuilders who really care about the muscle gain eat leucine-rich protein source. And studies on leucine sensing in human culture cells tell us there's a cytosolic leucine sensor whose name is sestrin. And when leucine level drops, for sestrin, it will go ahead and inhibit mTORC1. And when leucine is available, sestrin will bind with leucine, leaving mTORC1 being active. And sestrin is very conserved from human to flies. But unlike mammals who have actually 3 sestrin genes, flies only have 1, which really helped those in vivo studies. So here we chose flies to study the physiological role of this leucine sensing pathway. So we generated 2 sestrin mutants. One is a complete knockout, and we validated that even when leucine level is low in this mutant, mTORC1 is still active. So this mutant can mimic a constant leucine-rich state. The other is a homozygous leucine binding deficient mutant. And we picked a hydrophobic residue that coordinates the leucine side chain in flies based on the structural study we have done for human sestrin. And then we made this L431 mutant and validated that even when leucine level is high in this mutant, mTORC1 activity is low and insensitive to the changes in leucine. So for this mutant, it can mimic a constant leucine poor state. And then we moved to characterize the physiological phenotypes. So we first tested whether sestrin can help animals adapt to a low leucine diet. And when we put adult female flies on an amino acid replete diet, wild type and sestrin null flies live for a very similar amount of time. But on a leucine-free diet, sestrin null flies show a significantly shortened lifespan, which really suggests sestrin is very important for flies to adapt to and survive on diets low in leucine. We then asked if flies also require sestrin to detect food that is poor in leucine so they can actively avoid it. And to do so, we really need an assay to test if adult flies prefer eating leucine-rich over leucine-poor food. And we chose fresh apples as the base food. because they are very easy to cut and they're carbohydrate rich and protein poor. And it actually allows us to set up food choices that have different amino acid compositions, but the same content of complex sugar. So the food preference as it looks like this, we put age synchronized flies in a bottle containing 2 identical pieces of apple. The first one painted with a solution of 1 or more amino acids, and the second one with an appropriate control like water. Each also contain a trace amount of a unique DNA oligonucleotide, which serves as a barcode for measuring the consumption of each piece of apple by running qPCR on the whole fly lysase. And then we took the ratio of 2 oligos as our preference. And we first gave the wild type female flies a choice between leucine coated apple versus water coated apple. And y-axis here is the kind of preference fold difference comparing leucine apple and water apple. And x-axis here shows the amount of time the flies have been staying in the choice bottle. And you can see that they actually prefer to eat a leucine apple, but this preference does not show immediately. It emerges after the flies have been eating the apple pieces for around like 6 hours, and it increases to 5 to 6-fold by 24 hours. So this really suggests that the sensory cues like the taste or smell are unlikely to be the dominant reason for this leucine preference that we saw here. And I think it's very interesting that actually neither sesronol or the L401 mutant we generated, both of which have leucine insensitive mTORC1 signaling, showed a preference for leucine. And then we also asked if flies can distinguish between food with a more subtle choice, like the apple coated with 20 amino acids versus just 19 of them without leucine. And as you can see here, the wild type flies prefer 20 amino acid coated apple mildly, and this preference was absent in both sesron mutants. So now we know that the whole body mutant for sesron show a leucine detection defect. But we really want to ask what tissue actually account for this leucine preference. And to answer that, we knock down sesron in individual tissues by expressing a sesron shRNA under the control of various cell type specific GAL4 drivers. And we found that the sesron knockdown in glial cells was sufficient to reduce the leucine preference. But knocking down sesron in many other tissue, like the fat body shown here, has minimal impact. And then to see the expression pattern of sesron in fly glial cells, we mined a single cell RNA-seq data set for Drosophila brain. And they show that almost all glial cell subtypes, which are positive in the ripple expression, which is pan-glial transcription factor, also express the sesron mRNA shown in red. We then asked how does mTORC1 activity looks like in the brain, especially the glial cells under various diets. And we try to examine mTORC1 signaling by using a line expressing a GFP-based reporter for the MITF transcription factor, which is a Drosophila ortholog of human TFAB, a substrate for mTORC1. So MITF is suppressed by mTORC1. So upon mTORC1 inhibition, MITF's transcription factor activity increases because it goes to the nucleus more and it promotes the GFP expression. So our prediction here is like lower mTORC1 activity, a higher GFP signal you're gonna see. And in the wild type flies, we image the whole fly brain and look for differences comparing the control diet and also the leucine-free diet. And starvation for leucine really promotes the MITF activity in the group of cells very close to the esophagus. And it's a very interesting area because when flies eat food, it actually goes through the esophagus and then this area through the brain and then to the stomach and then gut. So it's very cool to actually speculate if there are some sensing for the nutrients happen here locally. And so now I'm showing you a zoomed in look for the esophageal area labeled by the blue circles. So the wild type flies show an increase of GFP positive cells upon leucine starvation. And the majority of these cells are actually glial cells indicated by the cold stain of the glial marker. But in the sesron null flies, fewer cells are GFP positive. And in the sesron L401E mutant flies, GFP positive cells were present even in flies fed the amino acid replete diet. And then we quantify the ratio of the GFP positive over total number of glial cells. Each dot here is an individual brain, while the wild-type flies show leucine sensitive changes of the GFP cell ratio. You can see sestrin null show a constantly low ratio, and sestrin L1E mutant had a constantly high ratio, which is consistent with this 2 mutant being leucine insensitive. So with these data in hand, we're trying to figure out how this one nutrient sensor regulates a complex behavior. Here I'm like showing a very simple model, even though we know very little about many steps in this model. So far, we have pretty good evidence showing sestrin in glial cells can respond to the dietary leucine level changes to regulate its mTORC1 activity in the cell autonomous manner. And then we speculate that mTORC1 activity change might lead to alterations of some secretory factors like ATP, serine, or some other molecules that have the capacity to impact the activity of the downstream neurons. In this case, we speculate the reward circuit might be involved based on some literature. And we really think the flies need to couple some kinds of sensory cues like the position or taste or shape of the apple pieces with the signal from the reward circuit to help them figure out which apple piece actually has the more kind of nutrients. And I have shown you the feeding behavior changes. There could be more types of behavior that we haven't developed assay to observe yet. But when mTORC1 activity in glia is locked in either off state due to the L1E mutant or on state due to not having sestrin in the flies, the reward circuit will also be constantly off or on without a dynamic change. We actually think the behaviors don't seem to appear to actually develop properly. And I think many cool questions remain here, such as the identity of the glial cells and the neurons, and also how the crosstalk happened between them two. And I think a potential approach that can really advance my work further is to develop a method that allows rapid isolation of rare cells from a complex tissue like brain. So I can do the metabolomics to see the change of the leucine levels upon various dietary treatments, as well as the transcriptomics to identify the potential protein markers that enable further kind of very specific manipulations for in the very genetic manner. So I think that approach will be very helpful for me. And with that, I really want to thank all members from the Sabatini lab and also my collaborator from Paramount Lab and Local Cell Lab, my thesis committee, and Star Market for providing me the apple pieces for this study. With that, I'm very happy to take any questions if there are any. Thank you.
Well, that's really terrific. I should point out that I became very curious about different interests in particular foods, and watching my wife as she became pregnant on 2 different occasions, where suddenly her interest in foods shifted from one type of food to another type of food. And I assume there must be some craving for something that's necessary during rapid child development to fulfill those needs of the child. And was very curious about how that worked. So I think this is a terrific insight. I would have never guessed it was the glial cells that might be providing that feedback to the brain that you're in an environment eating the right thing, and therefore you're happier in that environment. So it's I think a real breakthrough. It's very exciting.
That's kind of interesting because I mean, I think for the glial cell link, it's actually like, because that's kind of a cell type that's been neglected for a really long time. And people always think it's either neurons being directly having some feedback from the gut cells and then going up. And there are some like other studies showing there are some like gut cells being involved in the protein kind of preference type of phenotype. So I think there is a crosstalk right there. It just seems like the mTORC1 activity, those sensors don't seem to be really important for the gut cell feeding back to the brain. That's like from what my study has been. But maybe for other type of nutrient other than leucine, that would be the case. We don't really know yet, but yeah. And also like another thing I need to point out, I think this is currently the study in the flies, and we're actually very curious to see whether this is also the case in mammals, because we also have the sestrin knockout mice in the lab, and we're going to see whether this very similar type of regulation is conserved in a higher organism.
So there's a question from Piwa Lee. How the neuronal plasticity guide the TORC1/2 trafficking from glia cells? I'm not quite sure. I guess it's really about how the glial cells are talking to the neuronal cells. I think is what the question's about.
Yeah. So that's actually one of the kind of area that we're very curious about as well. Like, how do those two critical cell types are cross-talking to each other? There are some previous reports from Mark Freeman lab, who's like a dedicated fly kind of glial lab, and then they have been showing that the ATP can serve as like a very critical communicating factor kind of between these two cell types, that we're actually trying to figure out, because we really need a genetic marker to label those glial cells, and then trying to figure out the changes happen in those cells specifically upon the very different type of diets. And then we can start to isolate those and then figure out which molecule is actually more important for communicating with the downstream neurons. But yeah, that's a very interesting question we're actually pursuing.
Good. So I think at this point, we should move to the general
Hey,
discussion.
Lou,
Yes.
we have one question from Gary. Go ahead,
Oh,
Gary. Why
okay,
don't
Gary.
you
Yeah,
ask your
quick thing. That was a beautiful story. I'm just curious. I'm sure there are many other possibilities, but I'm curious if you tried manipulating that transcription factor that you're using as a reporter. Did you try manipulating it directly to see if there's an effect on this behavior?
That's an interesting point of view, because MITF is one of like many substrates of TORC1 in flies, and then so we haven't really done that yet, but we do have the shRNA targeting MITF, and we have that fly strain available already, and then we're like trying to actually knock down MITF in the glial cell, basically in the panglial cells right now, and just to specific to see whether it can impact anything in the downstream. But that will be a very interesting possibility that could be actually written like through the regulation from this transcription factor we have been using. But yeah, it's a very interesting experiment. Yeah.
Very nice. Yeah.
Yeah, the beauty of this model is that since TOR responds to lack of numerous amino acids, not just leucine,
yeah,
the exact same glial pathway could be independently used by all of them and still result in a reward for finding a food that supplies that particular amino acid that's defective without having a totally different circuit in every case.
Yeah, Lou, that's a very interesting point of view. And also, for all of those different type of sensors that our lab is still trying to figure out what they are. And right now, I think this kind of phenotype may also be very restricted to like where those sensors are actually expressed. For sestrin, it's quite interesting. It's actually highly expressed in the glial cells, but we're not very sure whether other sensors are also expressed in those cell types specifically, or they have other cells that are they actually need to respond to in those locations more. But I think that's a very interesting possibility in the future, that if we find more conserved sensors, then we can go ahead and pursue more.
Yeah, Kevin Klatt has a question which I think you've already answered. Do you think an analogous system exists for methionine via SAMTOR? I think you would argue it's the same. It's working the same way every time, right?
So for SAMTOR, it's definitely one of the things we're trying to figure out a little bit more. And in mammals, it's definitely one of the very important sensor for the methionine level, which is the SAM level basically. And then in the flies, I think the story gets a little bit more kind of complicated, because SAMTOR needs the KIX-TOR complex to bind and then to function for mTORC1 kind of activity regulation. But in flies, the KIX-TOR is not there. So we're trying to figure out what exactly is happening at the molecular level in the fly system for the methionine sensing right now. But yeah, I think that's an interesting possibility.
Yeah. So let's bring this to a more general discussion then. And maybe I want to start with a question for Joanne. in that, her technology has been really important in understanding how nutrients get metabolized into various tissues and into the bloodstream. But, I would just raise the issue, which I don't think has really come up in this meeting, about the kinetics of metabolism or of digestion, as opposed to the thermodynamics. We all assume that the total amount of food you eat ultimately affects how it's processed or where it goes. But in fact, we know that different foods are digested at different rates. And in particular, I'm talking about highly processed food, where you take exactly the same calories, the same composition, but you put it in a blender and it now becomes rapidly processed in the small intestine and absorbed. In particular, the carbohydrates getting into the bloodstream rapidly, causing a rapid increase in insulin. While if the same food of exactly the same calories, same composition, but not in a blender, but rather digested by the normal process of chewing, might take 3 or 4 hours to be digested, as opposed to 5 or 10 minutes, and therefore will have a very different effect, particularly on insulin, but perhaps on other hormones as well. So, Joanne, this is directed at you. Do you think that this is something we really need to discuss in more detail? And you're on mute right now, so.
I'm really not an expert in GI physiology or this specific thing, but what we have is a tool, right? Yes, and I know in the whole food plant-based community, there are people talking about whether or not, there's a big difference. There's a difference whether you eat beans or you eat tofu, eat soybeans. Because when you eat a whole bean, a lot of it goes very far down in your gut and feeds the gut bacteria. When you eat more processed food, less and less of it goes further and further down, and more of it can be absorbed more quickly. But there's what we're trying to do is provide the technology to answer these questions, because I don't think it hasn't really been studied, and it's a huge issue. Obviously, we know that highly processed foods, and if you can almost eat sugar water or white bread, and it's about the same, isn't it, on your blood glucose? It's very fast. So we know some things very clearly, but there's just so many metabolites, and there's food, not just glucose or sucrose. These things are all in foods, and the fiber, et cetera, that you consume with the food. It's really a fascinating subject, and we just need better tools to get to this data. So by taking lots of plasma samples, following labeled food, maybe you can get some insight into some of this, and you may find things, like we were thinking of short chain fatty acids the following day or so after eating heavy lipid labeled foods, things like that. You'll see those sorts of things.
Yeah, we talk about change in diet that's occurred to explain obesity in America. I grew up in rural West Virginia in the 1950s. Spent a lot of time with my grandparents, who were totally sustenance farmers, so they grew everything they ate. My grandmother did not have a blender, and she did have a stove, so she could cook food, but pretty much the food we ate was not processed at all. And there was no sugar at all because it was too expensive to buy, and you couldn't grow sugar cane in West Virginia. So it was virtually no sugar in the diet, and nobody was obese. And then West Virginia went in a very short period of time from the skinniest state in America to the most obese state in America over a period of about 15 years. And the biggest thing I saw change was the introduction of really, really cheap sugar, so that everybody was drinking Coca-Colas when nobody could afford to buy one in the 50s. Now they were cheap and everybody was buying them, and many other processed foods. And of course, less sustenance farming and more buying highly processed foods. So I blame that more for obesity than almost anything else, just from my own experience. But I would love to see other people weigh in on this, because that hasn't been a major discussion by nutritionists in America. there's a subset of people who are convinced of that, but it's not a majority. In which case, is it just lack of exercise? Is that why everybody's obese? I would argue no.
No, I would say no.
Yeah, I mean, diets definitely play a very important role here. Seems like Jennifer has a question.
But can I insert something? One thing I would say is exercise. The benefits of exercise are not restricted to the effects on energy balance and obesity, diabetes. That's all a very important aspect to it. But you know, even if people maintain normal body weight, normal body stature, the evidence is incontrovertible that exercise has other health benefits.
Yeah, I'm not arguing with that. Jennifer, you had a question.
Yeah, my question relates to the gut microbiome and how it might be intersecting in all of this. I mean, in the case of what you're talking about, Lou, if you shift to a more highly processed, sugar-laden diet, that's going to change the microbiome, which could have huge implications in terms of the metabolites that it's producing that then that connect to brain activity that in turn might drive some of the metabolic processes that we're talking about. So that's one issue that I'd like to hear people talk about as a part of the bigger theme of nutrition. But also I just wanted to make a point about the types of foods that people were talking about in terms of, you know, whether you blend it or I think and there's a whole field of food combining where, you know, you shouldn't be eating, you know, heavy meat with a lot of carbohydrate, and partly because the two different types of foods, when they get to your stomach, trigger different enzymes that change the pH in the stomach that allow for particular digestive enzymes to work more or less effectively. And so that's another big issue, I think, in terms of nutrition in relationship to the American diet, is that we almost always are combining, you know, French fries with hamburgers and buns, which makes it very hard to digest that meat, the hamburger and the meat. So that's sort of something also I think that, you know, could be considered in terms of nutrition. You know, I think classically people ate foods pretty differently.
So Jennifer, I completely agree with you, and I think Joanne already to some extent addressed this transit time to get to the colon, where you would have the microbiome helping in the digestion or playing some role, where with rapid carbohydrates, they all get absorbed very quickly in the first, you know, 10, 20 centimeters of the small intestine very often. But you know, the other thing I just want to point out is we talk about high-fat diets in mice, and then we also talk about ketogenic diets, which haven't really been discussed that much at this meeting. If you ask what's the difference between a ketogenic diet and a high-fat diet, the difference is the carbohydrate, not, you know, the high-fat diet and the ketogenic diet are both mostly fat. The ketogenic diet is about 80% fat, about 12% protein, and 8% carbohydrate. But the carbohydrate is very slow release carbohydrate. It's not sugar. It's lettuce, things that are digested slowly, green vegetables, etc. And that turns out to be a very effective way to lose weight. If you replace that with rapid release carbohydrate, you actually gain weight on essentially the same diet. And that's what we call a high-fat diet in mice. We just pull out that carbohydrate, rapid release carbohydrate, from the high-fat diet that we feed our mice. They will lose weight rather than gain weight. So I think this is something really that hasn't come up in this meeting, but we should really think about that.
Yeah, I'll just say one quick thing related, but then I know there's a lot of other hands. I mean, if you introduce sugar really, really fast, the cells can't drive it through, they can't use it as effectively, and they'll just shove it, you know, into citrate and into fat building. So
Right.
I think that's probably what's going on.
Particularly if it's fructose.
Exactly.
Very
Yeah.
effective.
So that's, you just, and that fat goes into the quickest tissues it can find, which would be visceral fat.
Deb, I think were you next, Deb?
Yeah. Yeah, so I just wanted to go back to something that you said, because it, really struck a chord with me, and it was just talking about the impact of the introduction of sugar into our food supply and the introduction of Coca-Cola. And so to my view, I completely agree that the combination of sugar, heavily processed foods, Joanne said these are toxic foods, and the fact that the food industry, they're pretty sophisticated, right? They're formulating these foods with the right amount of sugar and fat and the perfect texture, and they can pack all these calories into a small little bar that people can consume quickly. And so I think we all recognize that is probably the major driver in terms of the obesity epidemic, is that processed foods and how cheap they are. And so what I often think about is, that's not really what we study, right? We study biology, we study mechanisms, and but how much responsibility does this group of experts have in sending that message to the broad community, that these heavily processed foods are engineered to light up your neurons, make you addicted to the food, and they're going to have a detrimental effect on your health?
Yeah, I'm just saying that, one of my colleagues, Marcus Goncalves, who's an endocrinologist, trained in my lab, is now an assistant professor at Weill Cornell, has a paper that came out of our collaboration that's just coming out, I think, tomorrow in Nature, actually. And the paper shows that the fructose component of the sugar that we eat in processed form expands the length of the microvilli in the small intestine. And if you leave the fructose out, that doesn't happen. And we get to a biochemical mechanism by why it causes the villi to be longer. But the consequence of having longer villi is you absorb more food. So two people who have could eat exactly the same food, and if one person has been on a sugary diet for several weeks, their microvilli are longer and they just absorb more food. This is clearly very clear. This is seen in humans as well, I should say. But our study was in a more controlled environment in mice. So that's something nobody's really thinking about. Yes, Chris.
Yeah, just had my hand up a little while, so this will seem like a time slowed down, but getting back to the idea of the eaten food versus the chopped up and digested food, pre-digested or pre-homogenized food. Something that, again, we can't forget is the endocrine component, the incretin hormones that are secreted from the gut. So the further a meal can go down the gut before it starts to get absorbed, the more chance it has to encounter the microbiome. The microbiome produces a number of different metabolites, such as short chain fatty acids. And then you encounter special incretin hormone producing cells, which are nutrient sensing cells. Okay, so it would seem that maybe the fuel stimulated insulin secretion response is a rare example of metabolic fuels regulating an endocrine substance, but that's not true at all. All through the gut are fuel sensing cells that secrete other peptide hormones that have a critical role in how we dispose of fuel. And indeed, GLP-1 is very famous, but GIP, gastric inhibitory polypeptide, may be potentially the more important gut-derived incretin regulating pancreatic islet function that is coming onto the scene. So let's not forget the endocrine component, and that's a continuation of a theme that I was on yesterday.
Yeah, I completely agree with that. And it comes back to the bypass operation and the consequence of that, which you touched on earlier, which I think is, that's kind of what got me thinking about it as well. Huge effect of the bypass operation that everyone assumes is because the stomach doesn't want to contain as much food, but in fact, or doesn't digest it as well. But there's an endocrine component to that. I think that's very important in that bypass. You agree on that, Chris?
Absolutely. And in fact, Dave D'Alessio, my colleague, chief of endocrine and member of our institute here, was one of the first to show that bariatric surgery causes GLP-1 levels to go flying high. And that's a big part of how you can so quickly reverse diabetes with bariatric surgery.
Yeah. We have a question from Ron.
Yes, I want to follow up on the point that Debbie made that the dietary habit and probably the lifestyle is clearly a culprit of obesity pandemic, and the research in the community have shown plenty of evidence. The processed food is bad. The sedentary lifestyle is bad, but also like what time you should eat and the circadian rhythm all plays a role. We made a lot of discovery here. I think we have more to do and more to think about. But probably we should also think about what we make use of those knowledge. And currently, we're trying to encourage people to go back to the good living habits. Exercise, eat whole food, all of this. But we probably should also think that we should make the human beings more resilient to those bad habits. Like the bariatric surgery is one example, and trying to make ourselves, to modify ourselves to be more defensive. But we haven't done those at more molecular level. So this is a very provocative thinking. I think some people say I think backwards to try to accommodate those bad lifestyles. But that's how the modern life requires us to do. I mean, there's no more dinner at 5 p.m. anymore. And so there's no long break in the middle of the day. So maybe we should utilize the knowledge we learned at the molecular level to make human beings more resilient.
Thank you, Ron. I think we're coming to the end of our time limit here. So there's a break built in now between now and 1:35. So a 7-minute break.
Yes, perfect. Thanks so much, Lou, and everyone in this session. As Lou said, we will break for about 6 or 7 minutes until 1:35 p.m. Eastern.